I'm going to get started here by telling you how I got started, and I'll slowly be learning to work with the technology as we go through this. So I literally started out as a human in Yuma, Arizona. That's where I was born, and you know, lots of coyotes and other desert-dwelling critters and plants. This is pretty much what it looks like in Yuma. And if you look down at your feet, you're not going to see too much, maybe that seems very interesting. But if you look really close, there's lots of good stuff down there. But if you look off in the distance, you're always going to see some beautiful mountains. So that was how I grew up. And when I decided, when I was ready to go to college, I took the bold move. I got on Interstate 8 and went all the way to Arizona State University, which is incredibly giant now, but was moderately giant then. But I had some great classes. I got to take lots of courses in physiology and a couple of courses in neuroscience. And that's where I became very interested in how neurons function. I'm getting confused with buttons here. And like Steve Martin, I thought that I was going to be very interested in brains. And it turns out what I'm really interested in are the neurons, the cells of the nervous system, and how they interact with each other. So where did I go from undergrad? So with this interest in neuroscience, I'm going to sort of blow my cover here and tell you that I knew I was interested in neuroscience. I thought I would like to do research. Now, I didn't have any experience with research, not like now that you guys have. But I thought this is what I wanted to do because I was just so interested in understanding how things work in the nervous system. And my approach to looking for a grad school was basically applying to places I'd heard of. So that's a really bad approach, and I don't recommend that to any of you going forward. But I was incredibly lucky, and I ended up at a very wonderful place, and that was the University of California at Davis. And this is one of the egghead sculptures that's out in front of the library. And so there's lots of great deep thoughts and scientific inquiry that goes on at Davis, and lots of bicycle riding. So I don't know if you've heard about UC Davis, but it's covered with bicycles. And so I had a great experience there. That's where I first started studying the retina. I worked for Martin Wilson. We're a great match. And there was a small group of neuroscientists there. Now there's a lot of neuroscience at Davis. But at that time, it was a fairly small group. But it was a really sort of a nurturing group. And it was a great place to learn about neuroscience and get involved in studying the retina. Now, when I went on to do a postdoc, I was a little more informed at that point, so I think I made a little bit of a wiser choice. And I ended up at the University of California in San Diego in Nick Spitzer's lab, where I studied developmental neurobiology. And I studied how spinal neurons develop. So when it came time, though, to think about what I wanted to do in my own lab, when I was looking for a position, I decided that I was going to go back and get back into studying the retina. Okay, so that was kind of my first interest, and it turned out to be my long-term passion. So with that in mind, I was able to get my position here at LSU, and that's it. Okay, so there are a couple of approaches. There's sort of two main approaches to studying neuroscience. And one is that you can look at the disease state. And like Igor here, who's gazing intently at this abnormal brain. But the other approach is to look at normal function and study normal function and understand how the normal nervous system works. And that's the approach that I've taken in my lab. So in my lab, we studied the vertebrate retina. So I'm just going to give you a little bit of background on the retina. So the retina sits at the back of the eye. And usually when you see a diagram of the retina, what you see is a diagram that is basically of a cross-section of the retina. So I'm going to sort of build a retina here for you. We're going to start at the input side of the retina. These are the rods and cones, the photoreceptors that take photons and convert that into an electrical cellular signal. And so, so those are the rods and cones that do that. And this is the output side of the retina. These are the ganglion cells. And these ganglion cells gather all the signals that have been generated in the retina, starting with the photoreceptors, and send that signal to the brain, other visual centers in the brain. And in the gap here, it's not empty space, it's filled with interneurons. And so there are horizontal cells that signal in this outer region of the retina. And there are bipolar cells that connect the rods and cones with the ganglion cells. And then there's this third group of interneurons, and these are the amacrine cells here in blue. And these are the cells that I've been studying, and we've been studying in the lab for quite a few years now. So why amacrine cells? Well, let me just tell you a little bit about amacrine cells. So amacrine cells are very diverse. If you look at the morphology, there's over 40 different types. We really don't know very much about all the functions of amacrine cells, but we do know a few functions. And if you look at those, they're pretty interesting. And I'm just going to tell you about one. So to talk about this function of amacrine cells, I have to tell you about a certain type of ganglion cell. So remember, the ganglion cells are the output cells of the retina. And there's a class of ganglion cells that can do a really neat trick. And what that is that they will respond to a stimulus, light stimulus, moving in one direction, but not moving in the other direction. So how does this work? So here, let's say we're recording from an amacrine cell, making an electrophysiological recording, so we can see what the activity level is in the cell, and present the retina with a moving stimulus. So if we have a moving stimulus in that direction, this is a little too auto, in that one direction, you saw that the cell responded just by the red dot showing up. But if we had the stimulus moving in the other direction, we don't see a response. So that's the trick that the ganglion cell can do. And what we know now, lots of really clever studies, nothing that I've done in my lab, but in other labs, is that it's the starburst amacrine cell and the connectivity and the synaptic interactions by those starburst amacrine cells that give the ganglion cell their ability to respond in this specific way to the stimulus. So that's just one example, the kind of cool things that amacrine cells do. And as I said before, most of the cool things they do, we don't really even understand yet. Okay, so my specific interest in amacrine cells was to understand their synaptic function and how they're regulated, how those synapses are regulated. So we have lots of, this is just a generic slide with some neurons interacting, axons sending signals and making synapses onto dendrites, and this is just a blow-up of that. And so here we have a presynaptic terminal and a postsynaptic site. And basically what happens during synaptic transmission is that through the activation of voltage-gated calcium channels, there's a calcium elevation. And then there are lots of molecules that interact and that are completely absent from this slide, but there's molecular interactions, and then that leads to vesicle fusion, which allows neurotransmitters to be released from the presynaptic terminal and to bind receptors on the postsynaptic cell. These receptors are ion channels, so you have current moving and changing the response of the postsynaptic cell. Okay, so I'm really interested in how this works, and more so, we know the fundamentals of how it works, but how is it regulated? How do different sorts of molecules in the retinal environment affect how these synapses function? And so wouldn't it be cool if we had a way to sort of listen in on what's going on in this presynaptic area? And so, of course, I'm going to tell you that we do have a way to look at, listen to what's going on here. And so before I get to that way that we have of listening to synaptic transmission, I'm going to tell you a little bit about where these neurons that we work on come from. And they don't actually come from adult chickens. We dissect retinas from embryonic chickens and from chick embryos, and we take these retina, and we can make retinal slices, but I'm not going to talk about any of that work today. What we do is to dissociate the cells, so all of the retinal cells in the retina, we dissociate them, put them into a culture dish, and then over about a week, these retinal neurons develop and differentiate. And there are multiple different types of neurons, but we know how to recognize amacrine cells in culture. Okay, so we figured this out a long time ago. And these cells in culture act very similar to the cells that people have reported from amacrine cells in the intact retina. So it's a good model system for looking at synaptic transmission. Okay, so this is how we can listen to what's going on at the synapse. So here we have two amacrine cells in culture, and they appear to be have a physical interaction with each other. And these are patch electrodes, okay? And what you're looking at here is really what you would look at, what you would see down the microscope. What you don't see is that these patch electrodes, these little glass electrodes, are connected to amplifiers and all sorts of electronics that allow us to record the very small currents in these cells and to also to stimulate the cells. Okay? So what we can do in an experiment, one type of experiment we can do is we've patched onto a presynaptic cell and a postsynaptic cell, but we can't actually tell which is which, so we just test them in both directions. In our experiment, we depolarized the presynaptic cell to activate voltage-gated calcium channels, let calcium come in, cause release of neurotransmitter onto the postsynaptic cell. So we activate our voltage-gated calcium channels, presynaptic calcium goes up, a neurotransmitter is released onto these postsynaptic receptors, and these are ion channels, like I said before, and so we can record current moving through these ion channels in the postsynaptic cell. Okay, so like I just showed you, we can record from pairs of amacrine cells in culture. And if we do this kind of experiment, we see this sort of data. So these are just the currents that we're measuring in the postsynaptic cell. And so down here is showing, this is a voltage showing you what we're doing to the presynaptic cell. So basically, during this period of time, we're depolarizing the presynaptic cell. That's causing neurotransmitters to be released, and this is the current that's generated in the postsynaptic cell. So you see at the instant of the depolarization, you start to see postsynaptic current. You can tell that neurotransmitters are being released because we can measure the current postsynaptically. We can also tell something else from the data shown here, and that is that these synapses are GABAergic. And what that means is that the presynaptic cell is releasing GABA, and it's binding to receptors on the postsynaptic cell and activating the ion channels associated with those receptors. And we know that because the currents are completely blocked by bicupilin. So this is a compound that is well known to block GABA receptors, the specific type of receptors we have in these cells. So no postsynaptic currents of bicupilin, but after we wash it out, we get our postsynaptic currents back. So this is one way to look at synaptic transmission. Another way to look at synaptic transmission is just to record from one cell and listen for spontaneous neurotransmitter release from the other cells that are presynaptic to it. Okay, so here we just have one other cell, but there might be multiple other cells in the culture dish that are contacting this cell. Okay. So what do those sorts of data look like? And for those sorts of data, what we see is we don't see a big response because we're not stimulating anything presynaptically. But what we see is every so often a little deflection in the current. Okay, and each one of these deflections is called a quantal event. So what is a quantal event? This is so cool. Should have stuck a slide in there, but I didn't. So a quantal event corresponds to the current that's generated by the postsynaptic receptor when a single presynaptic vesicle has fused and released its neurotransmitter. So every time we see one of those little currents, we know that a vesicle has fused presynaptically. Now I need to move back. Okay, so this little current here is produced by neurotransmitter released from a single vesicle. Okay, so by measuring these little spontaneous currents, and we call them spontaneous because we haven't done anything to the presynaptic cell. Okay, we can, by looking at these currents, looking at the frequency, we can understand sort of what's going on presynaptically. Once again, these little tiny quantal currents are blocked by vicupulin, so we know that they're due to activation of GABA receptors. Okay, we can wash those out. So the data most of the data I'm going to show you today involves looking at these spontaneous quantal events. Okay, so we're interested in synaptic transmission between amacrine cells, and furthermore, we're interested in how molecules that are present in the retina that might be released under different circumstances. The flash just like froze my brain. Released under different circumstances might affect the function of the synapse. Okay, and so this is a cover of Science magazine from 1992, where nitric oxide was declared to be the molecule of the year. So we've known about nitric oxide for quite a while, but we're still finding out and learning more and more about what it does in the context of the nervous system and also the immune system and in regulating the vasculature. So we became interested in trying to understand how nitric oxide affects the function of synapses between amacrine cells. Let me tell you a little bit about nitric oxide. So nitric oxide is synthesized from an amino acid, from arginine. And there are a couple of enzymes, nitric oxide synthases, that are calcium dependent. And they lead to, they take arginine, these enzymes, and convert arginine into citrulline plus nitric oxide. So nitric oxide is a gas. It can act in the cell that generates it, or it can act in nearby cells because it can diffuse through the plasma membrane. And this nitric oxide leads to signaling events going on in the cell. And there are a couple of ways it's known to act. One is through soluble guanylate cyclase. That's an enzyme that generates cyclase GMP. Another is by directly modifying protein. So S-nitrosylation is a chemical modification of a protein that can change the function of the protein. And this is a reversible response. So those are a couple of ways nitric oxide can exert its effect on the cell. So why do we think nitric oxide is important in the retina? Well, it's been shown that nitric oxide is produced by cells of the retina. Specifically, these are amacrine cells in a living slice of chicken retina, adult chicken retina. And they're bright green because they are producing nitric oxide. So what we've done here is to load the retina with a dye called DAF, and the fluorescence of this dye is sensitive to the presence of nitric oxide. So when the fluorescence goes up, that means nitric oxide has been generated. Over here, we're looking at a slice of retina that's been treated with L-name that inhibits the enzyme, the nitric oxide synthase enzyme, and we also have this compound CPPIO. This is a nitric oxide scavenger. So if you do that, you don't see any gas signal. So we know this is from nitric oxide. So we've got nitric oxide being generated by amacrine cells. What is it doing to this thing? Okay, so here we are back at our synapse, and we want to ask this question of what nitric oxide is doing there. Okay, so the work I'm telling you about was done by a graduate student in my lab, Wes Maddox, and in talking about this research, I'm not really going to focus on these parts of the graph. This is what tells us that our data are statistically significant, but instead I want to focus on the current records, so they really tell the story. Okay, so if you patch onto an amacrine cell and it's connected to other cells, it's going to have a certain level. It's going to be receiving a certain amount of input just based on the release of cuanto from the presynaptic cell. So here we have a little bit of activity under controlled conditions. And then we add SNAP. What's SNAP? SNAP is a compound that generates nitric oxide. So once SNAP goes into an aqueous solution, it starts making nitric oxide. That's how we can apply nitric oxide, this gas, to the cell. And so this is all reporting from the same cell. Once SNAP is introduced into the extracellular solution, then we start to see a lot of these little downward deflections. I know they were upward in the other slide, but don't let that concern you. All you have to be concerned with is seeing these little quantal events, telling you that presynaptically vesicles are fusing with presynaptic cells, and we're detecting postsynaptic. So we get lots of more activity, a higher frequency quantal events, and that's mainly what we're looking at here. And then after you wash the SNAP out, things quiet down again. So spontaneous synaptic transmission, which is thought to be actually very important in the retina, specifically in the interplexiform layer where the amacrine cells signal, is increased in the presence of nitric oxide. So this experiment addresses sort of a postsynaptic question, and that is, well, it's kind of a pre and postsynaptic. We have quantal events without SNAP, but when we add SNAP, we have more quantal events. So are we activating release of the same neurotransmitter? Is this still GABA being released, or is some other neurotransmitter now being stimulated to be released? And so here we have our control cell, very little activity after SNAP, lots of quantal events. It looks very noisy because they're kind of co-occurring sometimes. And then when we add bicuculin, that blocker of GABA receptors, we see that everything goes dead quiet. So these, again, are GABA-filled vesicles which are fusing through synaptically. Okay, so we know that calcium is important in synaptic transmission, and so we wanted to look if extracellular calcium was going to be important in this process. So here we have a control cell. As you can see here, this cell has quite a bit of synaptic input. So a fair level of, a pretty good level of spontaneous activity. You take away extracellular calcium, and everything goes quiet. So no more quantal events. You add SNAP, and SNAP now can't really do anything in the absence of extracellular calcium. Okay, so this effect of nitric oxide is dependent upon extracellular calcium. And we can see that the synapse hasn't just shut down and gone home. The synapses are still active. We can wash this out. Okay. So we know that extracellular calcium is important, and that's usually because something is happening to let calcium into the cell. So we wanted to look to see if there was where was I? Yeah. So we wanted to see if there was, in fact, calcium elevation in the cell. And to do this, we do a different kind of experiment. So here what we're doing is that we're making optical results. So what we do is we have amacrine cells in the culture dish, and we've loaded them up with a fluorescent calcium indicator dye. And this is called Origin Green, so the one we're using in this experiment. And the way this works is that once the dye is in the cell, if calcium goes up in the cell, it binds to the dye molecule and increases its fluorescence. So here we have a measurement, and these are just a relative calcium concentration, not absolute calcium concentration. So we see that we have this baseline level of calcium in the cell, and then we add SNAP plus that nitric oxide scavenger that I mentioned a few slides back. And we see that there's not much change of calcium in the cell, but once we add SNAP without the scavenger, then we get calcium fixation. And these are average data from multiple cells all put together here. And the black line, if you wanted to follow that, just represents the mean. So nitric oxide does, in fact, cause calcium elevations. And somehow these calcium elevations are dependent upon extracellular calcium. So we know that calcium elevations occur, and we know that nitric oxide produces calcium elevations. So the next question we ask is the calcium elevation, the calcium going up inside the presynaptic terminal, really the key to this increase in spontaneous activity? So here we're back to recording from our postsynaptic cell. And what we've done is to use a compound over here called VAPTA-AM. And this is a compound you can load into the cell, and it is a calcium chelator. So what VAPTA is really good at is rapidly binding up calcium. So if calcium comes into the cell, then it will bind to VAPTA, and it won't be able to bind or interact with anything else in the presynaptic terminal. Okay? So we want to know if this kind of buffering of the presynaptic calcium will have an effect on the nitric oxide dependent increase in spontaneous activity. So over here we don't have VAPTA. We have a cell with a baseline level of activity. We add SNAP, and we get a big increase in the activity, and then we wash it out and it gets quieter. Over here we have cells loaded up with VAPTA, so we have some baseline activity. And so the VAPTA is going into the cell that's being recorded, but more importantly, it's going into all the other cells that are interacting with this cell. Okay, so it's going into all the cells. So with the presynaptic cells, with this calcium buffer inside, you see that SNAP not cause an increase in spontaneous events. Okay, it's limited in its ability. So, and here washing, we're just washing out SNAP, we're not washing out VAPTA, it's inside the cell. So this tells us that that calcium elevation is really the key to triggering the increase in spontaneous events. The interesting thing, though, is that it's not voltage-gated calcium channels that we know can trigger vesicle fusion and neurotransmitter release in presynaptic cells. So we've done experiments to rule out the voltage-gated calcium channels. So what we're working on now is identifying the sources of calcium. And so we know that we need to have a channel somehow on the plasma membrane that's letting extracellular calcium in. We think that there's also probably a contribution from internal calcium stores. So Wesley's working on experiments to dissect that out. And so I really just wanted to talk to you about this because just to give you an idea of the way you can approach a question about modulation or regulation of synaptic function by making these sorts of recordings. The other thing that we're looking at is also the effect of nitric oxide on the molecular, the protein-protein interactions that are also required for vesicle fusion. So with that, I will just thank you very much for your attention. And these are the folks in the lab this summer. Sahar Khan and Nisha Koshnandi are undergraduates, and we've got Vijay, Tyler, and Vernon all working away in the lab over the summer. Wes is the one, again, who did the experiments I talked to you about. So thank you very much, and be happy to answer any questions. Yes. So not, to be honest, no. So is there any connection with blindness in my research? And so, like I said, my research is very focused on the basic questions, the basic research. Blindness is often caused by this destruction of the photoreceptors. So there are a couple of ways that that works, and also damage to the optic nerve and ganglion cells. So the cells in the middle are not usually a target of in blindness. Yeah. Okay, so the experiments I talk about today were not in pairs. We were just recording from a single cell that was receiving input from multiple other cells. Okay, but when we do study pairs, what we want to do there is not so much, what we want to have is control. We want to be able to control what's going on in the so we would be asking different questions under those circumstances. But we want to have control of the presynaptic cell so that we can drive synaptic transmission at particular times or in specific ways. So by recording from two cells, that just gives you more control over the synapse, and you can actually tell the synapse to work or not to work. Does that answer your question? Okay. Well, they are two different experimental designs. So by measuring the spontaneous activity, we can count the basically the fusion events presynaptically, right? That's what we were looking at there with those little currents. So every time there's a current, we say a vesicle fuse. So we can count those. We can measure the frequency of them. When you do a paired cell recording and you drive the presynaptic cells to release neurotransmitter, then you're going to get a lot more neurotransmitter release. And so you can't resolve the frequency of those events because many of them are happening together. But you can get other sorts of information. You can study the relationship between voltage and the presynaptic voltage and the postsynaptic response. Lots of different forms of modulation you can study in that way. So it just depends on the question that you're asking. No, it has been seen in other cell types. It's been seen, for example, in hippocampus. Can you repeat it? The nitric oxide dependent increase in cytosolic calcium, has that been seen in other cell types? And I was just saying yes.
I'm going to give the remote site a chance to ask questions. So anyone at the Primate Center want to ask a question? Nope. How about New Orleans? Nope. Report. Y'all all are just too quiet. Any other questions here? Okay. Thank you very much.